So, welcome everyone, wherever you are joining us from, to today's Global Immunotalk. So, this is a wonderful initiative that was started by Carla Rothlin and Elena Zuniga and aims to bring all facets of immunology to a global audience from all corners of the world. And we rely on the goodwill of stellar speakers from all around the world who agree to give these talks. And today, we're very fortunate to welcome Peter Brodin. I should, just before introducing him, mention that this is in fact the last lecture of the present series. And so at the very end, we will have a little presentation about the Global Immunotalk. So please stay for that. So Peter is originally from Stockholm, in Sweden, and that's where he did his MD PhD training. And then he did a postdoc with Mark Davis at Stanford, where he did some seminal work that showed that variation in the human immune system is largely driven by non-heritable influences. So this is by comparing twins. And that was a really important study. Then after that, Peter returned to the Karolinska in Stockholm, where he is a professor of pediatric immunology. But more recently, he has also taken an additional position at Imperial College in London. In fact, he's joining us from London today, where he is the Garfield Weston Chair and Professor of Pediatric Immunology. And the focus of his work is really on developing and applying novel experimental and computational methods to describe the human immune system. And he's got particular interest as a pediatrician as well in the immune systems of children and early development of the immune system and how it then is regulated in health and dysregulated in disease. So as usual, the talk today will be followed by a Q&A session, which will take place in Twitter or X, and Peter will endeavor to answer your questions, and we'll have a little slide at the end with instructions on how to ask your questions. So in a minute, I'm going to ask Peter to deliver his lecture, but as usual in the series, I'm going to start by asking him a question before the talk, and the question, Peter, is, as a physician, why do you think it is valuable and important to also perform research? And in particular, why is it important for you?
Yeah, that's a great question. Thanks, Caetano, and thanks for the invitation. This is really an honor to give this lecture here in a minute. I think about this quite a lot because, as you might know, fewer and fewer young physicians and also other clinically active specialists take up a career in research. And this is driven by many different societal changes, economic pressures, etc. But I think we really need to think about the value of having a few at least of these individuals, such as myself, who combine these two activities. Because we want patients to get the best evidence-based care, and that requires research training to be able to identify what's the best treatment for a particular disease. But we also want people who have who are founded or grounded in the clinical reality and the problems that patients face to inspire even basic scientists and to sort of push the agenda towards novel and better therapies and understanding of disease. And I'm really proud to take up this role together with many other physician scientists. And for me, it's a constant inspiration for new scientific questions from these patients that I see and care for.
Well, thank you. And I think that's inspiring to a lot of both basic scientists and clinicians around the world. And it's wonderful to see this combination of clinical activity and basic research and ultimately potentially bringing benefit to patients. And so, Peter, without further ado, I'm going to ask you to start sharing your screen and to deliver your lecture entitled Human Immune System Variation and Development.
Very good. Thank you so much, Caetano. And I think you see the right screen now, which is in full.
So I think we might be seeing your presenter screen. So you want to just change.
Yeah, let me try to do that again. I'm sorry for that. Let me see. There are two here. So if I show this one, now you should see the presenter the same as before, right?
Correct.
But if I do this, now it should be the right one.
Yes,
No?
I mean we are seeing right at the bottom of the screen. We are seeing all the other slides as well.
What about
Um,
now? That's
we're also seeing that, but it's you can go
no, no,
ahead
no.
and
I have it properly. Let me do it right. I don't want to. That's really annoying when it's like that. So let me get it right from the beginning. It's this dual screen situation.
there we go. Now it's perfect.
Thank you. And thanks for the patience. So human immune system variation and development is the topic I'm going to discuss today. And I'm going to talk about some of our work in the last few years, some of the new and unpublished work, which is going to be the main focus. And before to set the stage here, I want to talk about this topic of systems immunology, because systems immunology is a topic that has been, gaining a lot of momentum lately, and a lot of attention is placed on this. But I think too rarely we define what we actually mean by systems immunology. To some people, it might seem like immunology studies with a lot of data. And to me, that's missing the point of what at least I think is valuable about systems immunology, because to me, it's more of a change of focus and a way of thinking about the immune system as an integrated system with many different specialized cell populations that cooperate both by stimulating and inhibiting one another, and collectively giving rise to an immune response, whether it's in an infection or a cancer or in an autoimmune disease. So it's more of a change of mindset than it is of practice, I would argue. And I think so. This is what we use in all of our studies, and what the sort of fundamental aim in what we try to do is to start by observing populations of patients. So, or whether it's, well, it could be patients, but it could also be children of different age, etc., to try to understand how changes occur over time or in between patient and healthy, etc. But ultimately generating hypotheses about how something is regulated or dysregulated from the human observation. And I think that's the fundamental shift that is really important in human immunology, as opposed to more reductionist mechanistic work, which is still really, really valuable and requires often model systems, but then, sometimes can lead us astray and get lost from the sort of human perspective. Ultimately, what we always strive to do is to understand something at the regulatory mechanistic level, whether it's a cytokine A that impacts cytokine B and feedbacks onto the initial production of cytokine A, as an example. This is where we ultimately want to go. And it's very difficult to go all the way from the human population down to this mechanistic understanding. But that's the long-term aim. And one of the things that is really powerful when it comes to human observation, given the fact that every single individual would be so different from one another as a consequence of genetic variability, environmental heterogeneity, etc., is to follow individuals with longitudinal series of blood samples, for example, or even better, tissue samples, but that's much more difficult to obtain. Because when following someone over time, changes that are coordinated between different components in the immune system would allow us to infer some of these regulatory relationships and generate hypotheses around these things. Now, for us, one of the real breakthroughs has been seemingly trivial and has to do with how we process samples. And the reason why this has been a breakthrough is because by fixing blood samples directly when they come out of the individual at the time of blood draw, we are minimizing the technical sources of variation, which actually is a real problem in a lot of human studies and really obstruct the biological changes that we care about. And so, first of all, by doing this sort of preservation, all of the cell populations, including the neutrophils, are preserved for subsequent analyses by mass or flow cytometry or other methods. That's good because it keeps the system more intact. But also the variations, which is shown on these graphs in the middle, the coefficients of variance is always lower in the whole blood situation that is preserved as compared to the PBMC preparation, which is the standard method used by most studies of human immune systems. So this is really important to minimize technical variation in order to see more biological variation. And this, as I said, might seem trivial, but actually has real important implications. And so for me, human immunology studies really took off while I was a postdoc with Mark Davis at Stanford University, as Caetano just mentioned, where we studied the variation in healthy individuals that were either monozygotic or dizygotic twin pairs. And we estimated how much of the variation that we observed in cell population frequencies, functional immune responses in vitro, vaccine responses, et cetera, how much of their variation were actually attributed to heritable as compared to non-heritable factors. And although there are specific traits that are very strongly determined by heritable factors, the vast majority of features or components of your immune system or responses that we measured were explained predominantly by non-heritable factors. So one conclusion from this is that our immune systems are really a sensory system for the environment in which we live, and they adapt accordingly. And so it's really important to keep in mind that we need to take the environmental component into account. And it's obviously a lot more difficult than studying genetics, for example, where we have excellent tools. But we really need to strive to do this. And then this, together with me being a pediatrician, led me to ask the question of when this environmental imprinting begins. And obviously it begins from the very beginning in fetal life, but this transition from the very protected environment in utero to the world outside at the time of delivery is arguably the greatest change in environmental exposure that any of us will ever undergo during the time of our lives. And so it seemed of particular importance to study what happens right around this time. How does a newborn child's immune system react to all of these new environmental factors? How does it adapt to them? And how do those factors shape our immune systems as that child grows up and goes about its life? And this is interesting also from the point of view that epidemiological data has shown that perturbations such as antibiotic use during the very first months of life have real implications in increasing risks of a number of different diseases, as shown from this review paper summarizing a few of these observations. And some of these diseases, I want to point out, like juvenile arthritis, can occur more than a decade after birth. But still, the risk is increased by antibiotic use early in life. So why would that be? So in 2016 or so, my team then at the Karolinska University Hospital and Karolinska Institute in Stockholm, with funding from the ERC, established a birth cohort, which is now over 500 children, healthy full-term children, as well as some preterm delivered infants, enrolled in studies together with their parents, and then profiled with respect to both blood immune system development with serial samples from blood, as well as microbiome colonization as measured by stool samples and metagenomics. And this has led to several studies describing the developmental changes that happen during the first few months of life. And I want to just talk about a couple of these observations we've made. First, several other groups have associated the presence of specific bacteria in the microbiome of stool samples with disease occurrence later in life. And here are just two notable studies, one by Arieta et al., looking in a rural Ecuadorian population and children with developing atopic wheeze, and this Vatanen study from 2016, looking at three populations in Russia, Estonia, and Finland with variable risks of autoimmunity and allergies. And both of these studies show that a higher abundance of bifidobacteria is associated with protection from disease. These are variable types of diseases, but they're all immune mediated to some extent. So high bifidobacteria early in life seems to be protective. In contrast, abundance of bacteroidea early in life seems to be associated with a greater risk of disease in both of these instances and in several other studies. So we were interested in digging a bit deeper into the mechanisms of this. And this is 1,050 metagenomes ordered by the day of life in which we've sampled them. And in orange here, you see the bifidobacteria and the actinomycetia, which dominate many of these microbiomes, particularly in breastfed infants, because these bacteria really thrive on human milk oligosaccharides in breast milk. Gamma proteobacteria we often think of as pathogens, but they are very abundant in some of these infants, despite being healthy. After the introduction of solid foods, which in this cohort happens between 3 and 6 months, these children show a diversification of the microbiome with a bloom of Clostridia. And these bacteroidea, which I mentioned early, are stably present throughout this period. They also bloom towards the end, but they are very variable between children. As you can see, some of these here show almost none of these bacteria present. So we zoomed in on bifidobacteria. I showed you earlier that this is associated with a benefit or protection from immune-mediated disease later in life. And one of the real interesting aspects of bifidobacteria is that many of their strains are efficient metabolizers of human milk oligosaccharides. That's why they expand in breastfed infants. But this also has been shown to lead to the production of metabolites that can modulate immune development. So in a study together with the company Infinite Health, we fed children, full-term babies, all breastfed in California, with either nothing or with a probiotic that contains a bifidobacteria that is optimized to metabolize human milk oligosaccharides. And what you see in these stool samples from the control groups on the left and this probiotic-fed children on the right is that the genes required for HMO metabolism are nearly absent in the control children, but very abundant in the probiotic-fed children, showing that these microbiomes are really taken over by these optimized Bifidobacterium infantis strains. And this gave us resolution to look at the immunological consequences thereof. And if we look at the stool cytokines, there's no significant difference from the beginning when they are first being sampled prior to probiotic treatment. But after 2 months of probiotic treatment, there are pretty striking differences in the cytokine patterns in stool, with much higher abundance of IL-17, IL-13, these Th2, Th17 type cytokines in the control children, while the probiotic treated are dominated by beta interferon. And we looked for metabolites associated with this, and we landed upon tryptophan metabolites, such as indolactate, which was much more abundant in the probiotic treated children. And to summarize this published work, we basically found that these beneficial microbes early in life are characterized by really efficient HMO metabolism, leading to the production of tryptophan metabolites such as ILA, but also a skewing of T cell populations, both in vitro and seemingly in the gut based on the cytokine patterns. And all in all, this was associated with lower levels of intestinal inflammation early in life, which we think is a beneficial thing. So we think that this is one aspect of a healthy immune microbe interaction established during the first few months of life. Now, since then, we've been trying to dig deeper into these types of associations because what we ultimately want to do is we want to identify the molecules that are involved in this crosstalk between the microbes that colonize infants and the immune system that needs to sense and decide what to do with those microbes. And one of the things we've done is from this cohort, we've delved deeper into a subset of children who are either fully breastfed for the first 3 months and have never seen formula, or children that have never seen breast milk and are fully formula fed during the first 3 months. And then we've looked by both stool samples and serum for metabolites using a complex 4 protocol metabolomic analyses, as shown here in the middle. These types of exercise leads to hundreds of thousands of features to be discovered, and only a fraction of them can be annotated at this point. But it gives us an ample set of candidates to look further at. And so what we've done is, if we just look at the separation between formula-fed and breastfed infants at 3 months using one of these protocols, so 50,000 features from the HILIC-positive stool samples, we see that they are very easily separated, which might not be so surprising because they're eating very different foods. But what's more surprising is that at 6 months, after these children have all introduced solid food, their microbiome has diversified, they remain different, suggesting that there's something that has locked into place that is probably associated with a health benefit in the breastfed infants more than in the formula-fed infants. And we're now looking at thousands of these candidate molecules with the support of Wellcome Trust through a discovery grant to really look at what the mechanisms of some of these metabolites might be in regulating things such as an LPS response and other types of immune responses in vitro. And there are many interesting candidates that we are currently working on here. Now, I'm going to switch gears a little bit, and I'm going to talk about a different project which is now unpublished but soon to be published, where we have looked at another facet of human immune variation. And this has to do with sex differences. And I became interested in this because if you think, everybody knows that human immune systems differ between males and females. But also from a life course perspective, there are changes that occur, as I already said, early in life with colonization of microbes. But there are additional changes that happen with sexual maturation and the new needs that the immune system now need to be able to tackle, such as sexually transmitted infections, for example, and in the case of a female, menstrual cycle, and eventually implantation and passive immunity, which are really important evolutionarily important processes that an immune system needs to cater to. So, and just to remind everyone online that sex differences are widespread and involve all kinds of different immune responses. It has to do with infectious disease severity. It has to do, and I'm going to give an example in a minute about COVID-19, vaccine responses, which tend to be stronger in females over males. On the other hand, autoimmune diseases are also often more common in females than in males, while cancers show the opposite pattern. And so during the COVID-19 pandemic, this became so evident. In the beginning, before we had vaccines, the case fatality rate was always higher in the males across a number of countries. And we learned through multiple groups and many studies that what really determines disease severity in the COVID-19 situation is the early type 1 interferon response, which seemed to be stronger in females, thereby limiting viral replication and leading to milder disease. Similarly, in children more than adults, this type 1 interferon response was stronger, associated with milder disease. On the other hand, in individuals that failed to mount a strong type 1 interferon response, more likely to be males, smokers, and other things, you saw a cytokine storm develop over time, which was mediated by TNF, IL-6, and other pro-inflammatory cytokines, as the immune system was fighting to catch up with this rapidly replicating virus. Now, what's true about all of these different sort of differences observed between males and females is that we know very little about the relative contribution of chromosomal differences and hormone differences. And this is the reason for the study, which I'm going to tell you about now, which was a study that me, together with my partner in crime here, Nils Landegren in Uppsala University, and also Ole Kempe and other endocrinologists working in Sweden in different centers, started to envision, which was a study where we wanted to monitor the immune system in individuals that are born, that are assigned one sex at the time of birth, but then undergo sex reassignment, or as the correct term is, gender-affirming hormone therapy, allowing them to obtain gender characteristics more in line with their identity. Now, we reasoned that we could profile the immune system over the course of this therapy to try to learn how hormones would alter your immune system, even when the genetic differences are not relevant because they are fixed. Now, with that said, I want to just point out that this is a study of people and human beings, and it's not a study of a model system. And this is a community which is under a lot of pressure across the globe in many different countries. And it's really important to keep in mind that what we need to provide for these individuals is evidence-based care. And we have very little evidence at the moment and very little knowledge about what the long-term consequences of hormone treatment is, both in trans men and in trans women. And I think we owe it to the community to try to figure that out and try to understand what the long-term consequences are. So I'm going to tell you about how the immune system is adapting in trans men receiving testosterone therapy. And I hope that this type of information will be useful in order to ensure the health and well-being of such individuals in the future. So in these individuals, testosterone is the only therapy provided. And what happens after receiving testosterone therapy is that the testosterone levels go from a reference range for female to a reference range in the male reference range level very quickly. And at the 3-month sample time, more or less all of these 23 individuals had met that criteria, and they were in the male reference range. Estradiol levels decrease rapidly as well, and so does progesterone levels. And in fact, when we integrate 8 different hormones, they all show a pattern, in this case, moving from right to left, which is shared among individuals, despite the fact that they are kind of scattered all over the place from the beginning and quite variable. So there's a common hormone reset, if you will, in these individuals. But what happens to their immune system? So this is looking at transcripts in a whole blood mRNA sequencing analysis. So what you see here are transcripts that are significantly changing as a consequence of testosterone therapy. And what you see in pink here, these are transcripts all associated with type 1 interferons, and they are suppressed after testosterone therapy. And on the other hand, transcripts associated with TNF alpha signaling via NF-kappa B, or the hallmark inflammatory response pathway, are induced as a consequence of testosterone therapy. So it seems as if the entire immune system shifts focus away from type 1 interferons and in favor of pro-inflammatory and TNF-mediated signals. The immune cells in the whole blood, which is preserved at the time of blood draw, is analyzed by mass cytometry, and in this case, 12.3 million cells measured by 50 parameter cytometry assays, and then clustered into the populations of interest. And what we found was, to some extent, rather minor effects, if you will, because there were quite few cell populations that changed significantly over time. But these 3 did, and the most striking change was a suppression in the number, or a decreasing number of plasmacytoid dendritic cells, PDCs, after testosterone therapy. There were also slight changes in mate cells and central memory CD8 populations. Interestingly, with respect to the PDCs, was that there was also a change in phenotype. And what this plot shows is an embedding, two-dimensional embedding of these, specifically the PDCs alone, showing a concentration in this phenotype here. And what that's characterized by is an upregulation of the marker CD81. Now, CD81 was not very well known to me on PDCs, but there was this study from Ed Engelman's lab at Stanford previously, which showed that CD81 expressing PDCs do not produce type 1 interferons when you stimulate them with TLR ligands such as CpG or R848. But if you culture them together with T cells, they support stronger T cell proliferation, as shown here, as compared to here. And those T cells that proliferate also are more likely to upregulate FOXP3, suggesting that both in mice and in human PDCs, that they might serve a different function from the classical PDCs that we know and love. Following that study, there was, however, additional granularity provided to the PDC knowledge or the understanding of PDCs that we had, both from the Viliani's group and from C et al., for example. There were multiple studies using single-cell RNA sequencing and detailed mass cytometry analysis, showing that there's a population called pre-DCs, which is different from the classical PDCs. They also express CD5. So we looked at this population, but we saw no change in these cells over the course of testosterone therapy. So we think that the changes that we see are actually mediated by the more classical PDCs. Now, what about function? So if we stimulate PBMCs from baseline or after 3 months of testosterone, and then we stimulate them with R848, and then we measure by single-cell RNA sequencing, what we find is that these prototype or canonical response genes are less strongly induced after testosterone therapy by individual PDCs. We can summarize this at the hallmark pathway level as a lower induction of hallmark interferon alpha and after testosterone therapy. Now, we also measured the proteins secreted using this sensitive Simoa assay in vitro together with Jamie Sugrue and Dara Duffy at the Institut Pasteur in Paris. And there was a trend towards a lower induction, although I think we were a bit underpowered in this analysis. Now, what might be regulating this PDC change in function? We looked at some of the well-known regulators, such as IRF7, which is seemingly decreasing in abundance at the mRNA count level in individuals after testosterone therapy, in individual PDCs, that is. On the other hand, SOX3, which is a negative regulator, together with SOX1, actually, negative regulators of the type 1 interferon response are increasing in transcript abundance after testosterone therapy. But what about other cells? PDCs are not the only cell type that respond to R848. We looked at monocytes in the same cultures stimulated with R848. And what we find is that, again, a suppressed hallmark interferon alpha response after testosterone therapy. But in these monocytes, we also noted something else, which was that the TNF response pathway was triggered actually stronger after testosterone, mimicking what we saw at the bulk RNA sequencing level. Well, this made us curious, and when we looked at not just R848 stimulation, but actually LPS stimulation and these monocytes, what we then saw was an even stronger induction of hallmark TNF responses. And if we look at some of these key genes, such as IL-1 beta, TNF itself, IL-6, we see that these are induced more strongly by LPS stimulation after testosterone than before. So this really suggested to us that there is some form of cross-regulation here going on between type 1 interferons on one end and TNF at the other end. And this was intriguing. And it was even more intriguing when we started to consider which cells actually express the receptors for the androgens AR or the estradiol ESR1 and 2. And it turns out from RNA sequencing data done by fact sorting cell populations and then doing really deep transcriptional analysis within the Protein Atlas consortium, which is a data set that my group provided in 2019, what we found was that the AR was highest expressed in PDCs, and so was the ESR2 receptor. And we confirmed this at the protein level using intracellular staining and flow cytometry, that PDCs express the highest level of the androgen receptor of all the major blood immune cell populations, suggesting that PDCs has a role potentially as a sensory or a sentinel cell for sex hormones. Now, the functional responses in the monocytes also led us to look in another direction, because one of the genes that we saw coming up after testosterone therapy in monocytes and T cells and NK cells was SLAMF7. And we became curious about, when reading this paper from Mike Brenner's lab, looking at macrophages from the joints of RA patients, showing that SLAMF7 was upregulated by gamma interferon in these cases. And when SLAMF7 was present, those cells always produced more TNF. It was a supercharge, if you will, of the TNF response by myeloid cells. And in our cohort, when we looked at baseline as compared to 3 months in monocytes, we found that SLAMF7 was induced to more or less all of the monocytes expressing this molecule, and the TNF response measured at the single-cell RNA sequencing level was always stronger by the SLAMF7 expressing as compared to the negative fraction of monocytes, suggesting that this might provide some mechanism, but not the entire explanation, I think, to the induction of TNF responses. Now, hormone levels, as I mentioned in the beginning, change in both ways. So testosterone is induced because we provide testosterone exogenously, but estradiol is also decreasing. So how can we decipher which one is actually explaining the changes that we see? And this is a real challenge and points to, or it illustrates the difficulty when doing human immunology studies. Because in vivo, there's no way we can just monitor our way to explain this. So we have to do some form of in vitro experiments. We thought about going into mouse models, but the hormone regulation in the mouse model is completely different. The whole biology around sex hormones is very different in mice than humans. So that was not an ideal option. Instead, we set up an in vitro experiment using human donor cells. So what we did was we took healthy female donors of reproductive age with normal levels of estradiol endogenously. We treated their blood, whole blood, in vitro with DHT, which is a form of testosterone that cannot be converted further into estradiol. Or we combined this treatment with the androgen receptor inhibitor to validate that the result was actually mediated through the androgen receptor. Or we try to mimic the loss of estradiol signal by blocking the estradiol receptor using fulvestrant. Or we had an untreated control. And what we found after LPS stimulation, looking at gene expression analysis again, was that NF-kappa B1, which is one of these many principal response genes in response to LPS, was induced by DHT. We lost that induction after androgen receptor blockade, and there was no effect of blocking the estradiol signal alone, suggesting that this is a direct effect of testosterone in an androgen receptor dependent manner. SLAMF7, on the other hand, was induced actually either by DHT or by blocking estradiol signals, suggesting that the balance between these two types of sex hormones might be regulating the induction of SLAMF7. And similarly, when looking at R848 stimulated cells, we found that the induction of SOX1, which is this negative regulator of the type 1 interferon response, was directly induced by DHT. And the same was true for SOX3, suggesting that this dampening of the type 1 interferon response might be a direct DHT effect through these regulators. we were a little bit surprised to see very minimal changes over the course of 1 year when it came to T cell populations. And I think the reason for this might be that these individuals are between 20 and 40 years of age. Most of their T cell populations are of memory phenotypes that have already differentiated. They're also mostly in the tissues, and we are looking in the blood. And so there might be some aspect of that not allowing us to see changes that might be occurring, because there have been numerous reports of sex differences when it comes to frequencies of different T cell populations, as well as changes such as exhaustion of T cells, which has been associated with androgens, for example, in prostate cancer, to name one example. We saw very little examples of this during the 1 year of therapy. What we did see, though, was that using a niche net analysis, which is a way to try to infer cell-cell interactions based on differentially abundant transcripts. In this case, we looked at IL-6, TNF, IL-15, and IL-12B, which are all induced in monocytes after testosterone. And then we looked at what might these transcripts, if they are induced, could they explain some of the other changes seen in other cell types, such as induction of granzyme, interferon gamma, and perforin in NK cells, as well as SOX1 in CD8 T cells, just to name some examples. And it turns out that we think those could explain some of these changes. And this, I think, was further corroborated when we did epigenetic analysis, looking at ATAC-seq data before, at 3 months, and at 12 months of testosterone therapy, which showed that in T cells and NK cells, there was an epigenetic increased accessibility, chromatin accessibility in binding sites for transcription factors associated with the NF-kappa B pathway, not with any of the others, suggesting that monocyte potentiation of the NF-kappa B sort of response genes was seen in T cells and NK cells. So that was of interest. And we looked a little bit closer at the NK cells, and we used the same type of setup with in vitro treatment of healthy female donor blood. And when we stimulated that blood with DHT and then performed PMA ionomycin stimulation to look at what cytokines these cells might release, we found, or mobilized intracellularly rather, we saw that DHT induced more gamma interferon by NK cells, particularly the CD56 DIM population. But that was not seen with ESR inhibition, suggesting that this was a direct DHT effect. And now you might wonder, well, this is still rather an artificial scenario for explaining normal differences between cis females and males in the context of COVID or other infections, as I mentioned earlier, because gender-affirming hormone therapy is absolutely a very special situation. Now, what Richard Forlin, who's a really talented PhD student in my group, did was he went to public data sets, and in this case, 2 sets of COVID data, COVID-19, PBMC, single-cell RNA-seq data sets. And what he found was that in SARS-CoV-2 infection, without any re-stimulation, the monocytes and the PDCs always produced lesser type 1 interferon transcripts in males than females. And for one of the data sets, the monocytes showed higher, or for 2 of the data sets, monocytes always showed higher hallmark TNF responses in males versus females, supporting the differences we've seen during testosterone therapy. Using a completely different data set, which is from this beautiful Lifelines cohort in the Netherlands, here looking at in vitro stimulated PBMCs, either by Candida stimulation or mycobacterial stimulation, Rickard found that the Candida induced lower levels of type 1 interferons in male PDCs as compared to female. When looking at the SLAMF7 expression on the monocytes, similar pattern was seen as in our scenario, which is that males expressed more of the SLAMF7. And when looking at mycobacterial stimulation, not only was there a higher TNF response in the male monocyte than the female monocytes, there were always higher levels in the SLAMF7 expressing as compared to the SLAMF7 negative monocytes in these cases, supporting that what we have uncovered during a gender-affirming testosterone therapy might contribute to explaining some of the differences between cis males and females. Now, I've told you that there's seemingly a cross-regulation between type 1 interferons and TNF-mediated responses. And I've illustrated this here as one side being potentiated by estrogens and the other side being potentiated by androgens, and that these two are cross-regulated in the immune system. And do we know anything about this from before? So it turns out that this cross-regulation might actually have been known previously. For example, if we provide anti-TNF blocking therapy to patients with inflammatory bowel disease, arthritis, etc., we know that about 15% of those patients will develop a lupus-like disease with increased levels of type 1 interferons, suggesting that blocking the TNF side potentiates the type 1 interferon side. There are also really fascinating case reports of trans men receiving estradiol. There is one case developing a lupus-like disease after receiving estradiol. And on the other hand, what we've shown is that the TNF response, which is potentiated by androgens, in monocytes especially, might trigger an adaptation in the T and the NK cell pathway, inducing stronger gamma interferon responses, which then feed back through this upregulation of SLAMF7 and potentially other mechanisms on the monocytes, leading to a further enhanced pro-inflammatory drive triggered by male sex hormones, androgens. Now, why might this be? So it's tempting to speculate from an evolutionary point of view. And what we know is that TNF-alpha is associated with pregnancy complication and failure to implant a fertilized egg. That suggests that during pregnancy and in the menstrual cycle, allowing for successful implantation might actually require suppression of TNF-alpha mediated responses. At the same time, type 1 interferons, which are not detrimental to pregnancy success in terms of implantation, might serve as a really important defense against vertically transmitted infections, and thereby might be beneficial to boost in the situation of a pregnant female. What about the males? Are there any benefits to the other side of the story here? And from an evolutionary point of view, but also within the framework of life history theory, which deals with the fact of prioritizing limited resources for different traits, such as reproduction or growth and maintenance, it's tempting to speculate that TNF-alpha might be boosted because it plays a really important role in muscle repair and growth. And we know this because after an acute exercise situation, local macrophages in the muscle tissue will mobilize TNF-alpha to promote repair and growth. In contrast, type 1 interferons can serve a detrimental function on muscle fibers. And we know this from patients with myositis, which is often a type 1 interferon-driven disease, and also from experimental work in vitro, which has shown that muscle fibers cultured in the presence of type 1 interferons will waste over time. So it is tempting to speculate that these cross-regulated type 1 interferons and TNF-alpha might serve important evolutionary functions to maximize the success of implantation and pregnancy in females, while allowing increased priority for muscle growth and repair in the males. And this was all I wanted to share with you today. I have to thank a phenomenal group of people that I'm really fortunate to work with, both current members and former members. I pointed out work by so many of these individuals, and I was trying to show on each slide who has contributed experimental work and who has contributed with computational data analysis work. So I hope that was clear throughout. I also want to thank my collaborators, especially Niels Landegren, for the collaboration on the gender-affirming hormone therapy study, together with his PhD student Fabian Sard and Ole Schempe, who are professor at KI in endocrinology, as well as endocrinology colleagues who care for the patients undergoing gender-affirming hormone therapy around Sweden and enroll these patients, as well as collaborators at Institute Pasteur, Dara Duffy and Jamie Segru. And for the initial part, looking at bifidobacteria, it was a very fruitful collaboration with Infinite Health and Bethany Hendrick in particular. And I hope we can have fruitful discussions on all of this work during afterwards here on Twitter. So thank you very much.
Well, thank you again, Peter, for a wonderful talk. Just to remind all of the audience that the instructions on how to ask Peter any questions that you may have about this work can be, the instructions are now on your screen. And so this will be all done after the talk via posts and replies to those posts. So it just remains for me to thank Peter once again for this great talk, a suitable end to the current series. And then I just want to invite Carla to put up this short video about global immunotalks. And thank you, everyone.
And I'm here with my dear colleague, Carla Brodin, with whom we founded Global IMMUNOTALKS in April 2020, already so long ago, Carla. And first of all, I would like to thank all the organizers for their generous effort to put the programs that we have been enjoying, and also all the speakers. And last but not least, all of you who attend Global IMMUNOTALK sessions, and also who has nominated many of the speakers that we have selected for the programs over the past year. Indeed, many thanks to all of you. Please help us spread the word and make Global IMMUNOTALKS more accessible to everybody across the world. And stay tuned for the upcoming really exciting program for the next semester. Thank you. Bye. Thanks. Bye-bye.
And so with that, we will close the session for today. Thank you again, Peter. Thank you, everyone, for participating. And see you again at the next edition of Global IMMUNOTALKS. Bye.
Thank you so much, Caetano.
Bye.